I asked to show how damage to cells in a finger could activate a nociceptor signal neuron, and then what's the difference between a minor pain and severe pain. So this sketch nicely shows a damaged cell releasing substances. They bind to a G protein coupled receptor that through that pathway opens the sodium channel. If enough sodium channels get here, we get a few action potentials per second, low intensity. High intensity would be more signals, so therefore binding to two locations, more sodium in, therefore more positive charge getting here. That would start earlier action potentials and sooner action potentials after each absolute refractory period and that would give you lots of action potentials per second. And that's how the cells that receive it would encode that as a high intensity sound. Another example with green being very low intensity, just one molecule binding, and with more you get all this additional charge here, more action potentials per second starting and going out to the branches. I wanted a mammalian cochlea with hair cells, how they function, a low frequency 100 Hz sound, and then the difference between a soft and a loud sound. Here's the cochlea unwrapped with the basilar and tectorial membrane in the middle containing the hair cells, and there would be a neuron here shown down here and also down here for the two examples. If this vibrates up and down, that vibrating does a little bending, does a little shear stress against that, so the hairs get bent. That opens stretch channels. And the sodium opens voltage-gated calcium channels, which bind to a calcium-binding protein, causing release of neurotransmitter. A soft sound is bending these only a little, maybe only a very little, and so you get less sodium in, less neurotransmitter release, and fewer action potentials per second. A loud sound is bending them more violently, so the stretch channels are open more and for longer. You get more calcium in, more neurotransmitter release, and more receptors bound by the additional neurotransmitter release. You get more action potentials per second. The simplest sketch that I graded was this one. I liked it quite a bit. Here's the magnetite crystal where we want a fish, and when it's pointing north, we want a neural circuit that would cause one neuron to be activated if the fish turns to the east and a different one if the fish turns to the west. I really wanted a magnetic circuit, but some people just graded it with the fish was pointing north, it happened to be pointing north, and if it turned left, it signaled one thing and right another. But this is what I had in mind. Here's the magnetite crystal, which is always trying to point north. And so if the fish turns a little bit to the east, so if it turns to the east, it's going to move that magnetite crystal, and that's going to push against the neuron and signal signal east. And then with more neurons here, the more it turns, it affects just one neuron or more. This showed it a little bit more clearly. So here's our fish. The needle is pointing north. If it turns to the east, then that crystal is going to try to actually go, if it turns east, the crystal would actually be pushing against this one. But the same idea counts. That deforms it, that opens stretch channel, sodium comes in, we get action potentials from that turn. A more intense signal would be if it turns further, it's going to push further against this and deform the membrane even more, open more stretch channels, so you get even more sodiums, and so you get more action potentials. Another version, if we turn one direction, we activate this cell. If it turns the other direction, it activates this cell. The further it turns, the more it stretches, more neurons in, more stretch channels open, and more action potentials per second. So in this case, action potentials per second would tell it how far it had turned. Last one was retinal, and I wanted you to think about iodopsins, different colors. They respond to different wavelengths of light, and what's happening in ways that are not completely clearly understood, at least as far as I can find out, is the retinal is held in different positions, probably with different tightness around that bend. For whatever reason, some photons get to it and some don't. I like this particular suggestion. So here's the overall retinal. Photon hits it, that straightens out from the 11 cis, the 11th carbon. Instead of being bent, it changes to all trans, so it's straight. That means it no longer binds properly in its binding site. G protein coupled receptor is activated. Over time, that will come back, but in the meantime, we've got a G protein here. It activates adenylate cyclase. We get cyclic MP, and then that cyclic MP opens channels. In terms of a mutation that might cause a change in the light wavelength it picks up, this answer suggested maybe red light is lower energy, green light is intermediate, high energy ultraviolet is more energetic than red light, and so maybe it takes more energy. So this is held in some way. We have a change in an amino acid somewhere that with these different bonds, it's maybe held in such a way that it takes this one will respond to a lower wavelength, whereas this one is held in such a way that it takes a more energetic wavelength. And that's a reasonable one. I saw other reasonable hypotheses, and I could not tell you which one is correct, and even if any of them are correct.